Root rot of conifers, that is one of the major forest diseases. The effect of the infection is that the bottom part of the stem gets rotted, which is strongly devaluating the wood. Root rot can also lead to wind throw, growth reductions, and mortality. Overall, in Europe, economic losses for forest owners are estimated to approximately 2 million euros per day. The major causative organism of the root rot is Heterobasidion species, and there are several in Europe. The fungus is spreading with spores that are airborne and can land on wounds and cut surfaces like stumps after forest operations. And this happens when the temperature is above 5 degrees Celsius or below 30 degrees Celsius, and when conditions are not too dry. And once the fungus gets established on for example, a thinning stump, it can grow down into the root system and via root contacts spread to nearby trees. And in the new tree, in the living tree, it can grow up for more than 10 meters in the stem, and it can also survive in root systems for decades. And you can control this spore spread and the new infections by spraying, for example, stump surfaces after thinning operations with a competing fungus or with a urea, for example. And these preparates, they will alter the biological and chemical environment in disfavor of Heterobasidion. Although stump treatment will to a large extent stop spore infections, a high proportion of conifer root systems are already infected in forests, and they provide inoculum to threaten future forests. And this is really hard. You cannot really reach these with the stump treatment, these root infections. So you have to find a way to stop the growth of the fungus. And this can be achieved by reducing the growth of the fungus in host trees with a more resistant genotype. And the spread will then be slower and the infections will be fewer in the forest. Testing for resistance is a tedious process in the field, and actual stem rot develops normally only after several decades. So you want to speed up the process. And this you can be doing by searching for and identifying genetic markers that correlate with lower susceptibility. These markers can, for example, be DNA sequences that code for host processes that are responsible for the resistance. Tests that correlate the resistance in the field are, for example, inoculations with Heterobasidion in young trees. And trees that resist fungal growth carry certain DNA variants that can be used for search for root rot resistance in any tree population, and thereby speeding up the finding of new resistant trees. The research at the Department of Forest Mycology and Plant Pathology at Swedish University of Agricultural Sciences, in collaboration with Skogforsk, has introduced and validated a set of genetic markers that are now introduced into tree breeding of Norway spruce in Sweden. Resistance testing in young Norway spruce trees, together with genetic methods such as quantitative trait loci mapping, QTL mapping, has indicated the phenylpropanoid pathway to be an important component of the resistance of conifers against Heterobasidion.
These trees are candidates for the next generation breeding material of Finnish spruce. They are being tested for a gene which gives tolerance against Heterobasidion and was found by Swedish researchers. So this is an excellent example of collaboration between Nordic countries. All organisms are parasitized by viruses, and that includes also the most important tree pathogens in boreal forests, such as Heterobasidian fungi.
Yes, more than 20 species of viruses are currently known to infect Heterobasidian fungi, and new high-throughput sequencing analyses are now allowing us to find even more viruses. Local diversity is also high, as many different virus species are usually found even within single forest sites. These viruses form chronic infections, as they are able to reside inside single mycelia for years or even decades. They may also transmit between separate fungal mycelia when they form connections.
This wealth of viruses has led us to think about the possibility that perhaps Heterobasidian viruses could be used in practical forest management to control their devastating tree pathogenic hosts. The first question arising in this context was whether we could find any viruses pathogenic to fungi.
That is a real problem because these viruses do not form infective particles like human viruses do, but reside only within fungal mycelia. On the other hand, diseased fungi are also very hard to isolate. As other fungi in these samples, they grow over them in artificial media used for mycelial isolations. And furthermore, these disease-causing viruses are found probably very rarely, as evolution is acting against them, because they depend so strongly on the host.
To understand the effects of viruses on their hosts, we need to create genetically identical strains with and without the viruses to be studied. That can be made by curing the original mycelium of the virus or by transmitting it to a new virus-free fungal strain. With this kind of comparisons, we and other researchers have shown that most fungal viruses do not cause diseases on fungi. However, a few years ago we found a virus causing a serious disease on Heterobasidian root rot fungus. In a mature spruce tree, the virus reduced the spread of Heterobasidion by more than 70%.
Yes, and that led to my doctoral thesis project where I found another disease-causing virus on the same fungus. In respect to biocontrol, these two viruses are able to support each other in spreading among different mycelia and also in causing a serious disease on the root rot pathogen.
Based on our observations in the laboratory, we understood that we have a pair of potential biocontrol agents against Heterobasidian root rot. A virus-based control based on those could, at least in principle, be used in normal even-age forests, but also in continuous cover forests where no other measures are available to mitigate an already existing Heterobasidian infection. And that's not possible even by breeding of tolerant trees, as the forest regeneration will be based on advanced growth instead of planted seedlings.
However, before we can save forests like this one, we need to solve several questions. For example, how to infect the disease-causing fungus by our viruses in the forest? What is the degree of resistance against the viruses in Heterobasidian populations? And finally, are there any risks in such a virus-based biocontrol?
These are exactly the questions that we address in our new projects. And from few years ahead from now, we will have the answers to them.
current view on the practical virus-based biocontrol is such that after cutting, a heterobacterian strain infected with viruses will be inoculated on the cut stump, showing symptoms of heterobacterian root rot. There, the virus will spread through the root rot, causing mycelium in the stump, root systems and neighboring trees, and thus colonize the whole mycelium, causing a disease on the fungus. As a result of this process, the heterobacterian strain will not be able to decay our conifers, and the forest site will be cured from the disease.